Hello, and welcome to this Microbiology Bio 203 video lecture on immunology and antimicrobial drugs. I'm Mr. Kennedy, and I'll be your guide as we briefly explore this topic together. The world of immunology begins back in the 18th century with the work of such men as Edward Jenner and Louis Pasteur. Edward Jenner is probably most well known for developing a connection between cowpox and smallpox, one of many diseases that ravaged human populations in this time period. Louis Pasteur, aside from developing the process we now know as pasteurization, also studied rabies. There are countless diseases that in the 18th century and beyond have been considered quite serious and have killed thousands of people. In today's day and age, we have nearly eradicated many of these diseases through the process of vaccination. None of these diseases has been driven all the way to extinction as a result of reservoirs of disease-causing organisms that are maintained in developing countries and in populations who are not vaccinated. Below is a list of disease-causing organisms and the vaccines that are used to prevent them. First on the list is diphtheria. The diphtheria vaccine is produced using purified diphtheria toxoid. The meningococcal meningitis vaccine is produced using purified polysaccharide from Neisseria meningitis. The whooping cough or pertussis vaccine is produced by using killed whole or acellular fragments of Bordetella pertussis. The pneumococcal pneumonia vaccine is produced using purified polysaccharides from 7 strains of Streptococcus pneumoniae. Purified tetanus toxoid is used to vaccinate against tetanus, and polysaccharides from Haemophilus influenzae type B, conjugated with protein to enhance their effectiveness, is used to vaccinate against Haemophilus influenzae. The flu, or influenza, has a vaccine that is administered and produced in 2 different ways. The injected vaccine is produced using an inactivated virus cocktail based on the strains most prevalent at the time. The nasally administered vaccine uses an attenuated virus with the same parameters. Attenuated virus vaccines are also used in measles, mumps, rubella, and chickenpox. Poliomyelitis and rabies both use killed viruses. Antigenic fragments of viruses are used in hepatitis B, and activated viruses in hepatitis A, live vaccinia virus in smallpox, and attenuated viruses in herpes zoster. Antigenic fragments of viruses are used for human papillomavirus. There are several types of vaccines available to us in immunological studies. Attenuated whole agent vaccines are vaccines created by reducing the virulence of a pathogen, but still keeping the pathogen viable or alive. The MMR vaccine is an excellent example of just such a vaccine. Inactivated whole agent vaccines are made from microorganisms such as viruses, bacteria, or other that have been killed through physical or chemical processes. These killed organisms cannot cause disease. An example would be the Salk polio vaccine. Toxoids, as mentioned previously, are a chemical modified toxin from a pathogenic organism. The toxin is no longer toxic, but still can evoke antigenic responses in the patient and can be used as a vaccine. One such example of a toxoid is the tetanus vaccine. Next, we have subunit vaccines. Subunit vaccines are made using a fragment of a pathogen, typically a surface protein, that is used to trigger an immune response, and stimulate acquired immunity. The acellular pertussis and recombinant hepatitis B vaccines are examples of subunit vaccines. Nucleic acid or DNA vaccines are vaccines composed of purified, closed circular plasmid DNA or non-replicating viral vectors containing genes that encode for viral antigens. West Nile vaccine for horses is an example of a nucleic acid or DNA vaccine. Next, we'll explore ELISA, or enzyme-linked immunosorbent assay. ELISA is a plate-based assay technique designed for detecting and quantifying soluble substances such as peptides, proteins, antibodies, and hormones. Other names, such as enzyme immunoassay, are also used to describe the same technique. This is an illustration of direct ELISA, where an antibody is absorbed to a well or delivered to a well. Then a patient sample is added, complementary antigens bind to the antibody, enzyme-linked antibody specifics for the test antigen is added and then binds to the antigen, forming a sandwich of sorts. The enzyme substrate is added and a reaction produces a product that causes a visible color change. In indirect ELISA, again, an antigen is absorbed or delivered to a well. Patient serum is added. Complementary antibody will bind to the antigen. Enzyme-linked anti-HSIG is added and binds to the bound antibody. The enzyme's substrate is added, and again, a reaction produces a product that causes a visible color change. Finally, let's examine antimicrobial drugs. Antimicrobial drugs do as their name implies and inhibit microbial growth or kill microbes outright. Chemotherapy is the use of drugs to treat disease. Antimicrobial drugs will interfere with the growth of microbes within a host. An antibiotic is any substance produced by a microbe that, in small amounts, inhibits another microbe. And selective toxicity. Selective toxicity is a situation where a drug kills harmful microbes without damaging the host. Antimicrobial drugs first gained recognition and hit their heyday in the early 1900s. In 1928, Fleming discovered penicillin produced by penicillium. And by 1940, Howard Florey and Ernest Chain performed the first clinical trials of penicillin, which would later prove to be what many considered a magic bullet or miracle drug. This table illustrates several organisms and the antibiotics that are used to treat them. Finally, with regard to antimicrobial activity, we need to consider that some antimicrobial drugs are broad spectrum and some are narrow spectrum. These terms can be used in the same regard as pesticides, which carry similar names. A broad spectrum pesticide, just like a broad spectrum antimicrobial agent, is effective against a large number of microbes and vice versa for the narrow spectrum. Antimicrobial drugs may either be bactericidal or bacteriostatic. Bactericidal antimicrobial drugs will kill microbes directly, while bacteriostatic will prevent the microbes from growing. This graphic lists a number of antimicrobial drugs and their mode of action. Note that some inhibit cell wall synthesis, while others inhibit protein synthesis. Others still inhibit nucleic acid replication and transcription. Others injure the plasma membrane. And finally, some will inhibit the synthesis of essential metabolites. Make sure you note which organisms are responsible for which mode of action. The effects of combinations of drugs, our final slide, synergism and antagonism. When taking drugs, it's important to take into account all medications that patients are currently using. Synergism occurs when the effect of two drugs together is greater than the effect of either alone, while antagonism is a situation where the effect of two drugs together is less than the effect of either alone. And this concludes our coverage of immunology and antimicrobial drugs.